Hello everyone, today we are going to discuss about the topic cultivation in culture medium. First let us see what is culture medium. So basically a culture medium refers to a media which supports the growth of any organisms. A nutrient medium may be a nutrient rich liquid or it may be a semi-solid medium. A culture medium has a various applications in biotechnology, biofuel industries, food production or in scientific research. All know a culture medium is used to support the growth of algae and it is enriched with necessary nutrients such as nitrogen, phosphorus and trace elements of other necessary nutrients. And now we are going to discuss about the various culture medium used for the cultivation of algae. First BG11 medium. It is used for the cultivation of cyanobacteria as well as green algae. And it is enriched with nitrogen, phosphorus, potassium, calcium, magnesium and trace elements of other essential nutrients. And it is used as a medium for the growth in fresh water as well as in marine. And next F2 medium. F2 medium is often used for the cultivation of marine algae such as chlorella and isochrysis. It is enriched with the nutrients like nitrogen, phosphorus and other trace elements and vitamins. Next, Bowles basil medium. It is used for the cultivation of freshwater algae. Chlamydomonas species is used for the cultivation in Bowles basil medium. And next, chlorophyte medium. It is used for the cultivation of green algae. And next, soil extract medium. This medium is used in the research laboratory to simulate the natural conditions for algal growth. And next let us discuss the steps involved in the cultivation of algae. First we have to select the species and then we have to prepare the culture medium, then inoculation for the algal culture, and then we have to monitor the regular growth conditions like water, light and temperature and pH. And it is cultivated in two types, open pond system as well as closed bioreactor system, and then harvesting of algae. Net Species selection. Green algae such as Chlamydomonas chlorella is commonly used for research laboratory and then as a food supplement and also for biofuel production. Blue-green algae is also known as cyanobacteria is used for biofuel production, water treatment and also as the source of food supplement. Diatoms, it is important for the carbon fixation and it also used in aquaculture. The examples are Phaeodactylum trichonutum. And then red algae, it is also used for the food production, agar extraction and cosmetics. Examples are Propyta glaciaria. Next, we have to prepare the culture media for the cultivation of algae. As we all know, algae require specific nutrients for their growth. The common media used for the cultivation of algae are BG11 medium for cyanobacteria, F2 medium for marine algae cultivation, and Bowles basil medium for the cultivation of freshwater green algae. So after preparing the culture media, we have to sterilize in the autoclave to reduce the contaminants present in the medium, and then we have to adjust the pH to 6.5 to 8. To reduce the pH we have to use hydrochloric acid, and to increase the pH we have to use sodium hydroxide. And then algae inoculations. Once we have prepared the medium and then we have to sterilize, and the next step is to inoculate the algae into the medium. Inoculate the inoculum into the prepared media. So we have to monitor various growth conditions during the cultivation of algae. The algal growth depends on light, temperature, pH during cultivation. As we all know, algae are photosynthetic organisms. They require light for their growth. On large scale production, artificial lighting system is installed for the light. Temperature should be maintained between 20 degrees Celsius to 30 degrees Celsius and pH should be maintained at 7 to 8.5. Types of cultivation system. First, open pond system. It is also known as raceway ponds. They are shallow and it is an artificial pond. They depend on the sunlight for their photosynthesis reaction. It is used for the biofuel production in large scale. And then Closed photobioreactor. In this, light temperature, pH, and aeration will be monitored very carefully. The photobioreactors are made up of glass, plastics, or stainless steels with a model of horizontal, vertical, tubular, and flat panel. And next, harvesting. Algae can be harvested once they reach a sufficient cell density, usually after several days to weeks. And first, flocculation. A chemical agent such as aluminum sulfonate iron chloride is added to the algal culture. This process clumps the algae together and lead to the formation of flocks, which can be easily separated by gravity. And next, centrifugation. Centrifugation by high speed is used to separate the algal cells from the medium. And next, filtration. After centrifugation, the cells are filtered through fine mesh or membrane filter. And next, microfiltration. In this, the algal cells are passed over very fine membranes with the pore size of 0.2 to 10 micrometer. And next, post harvesting process. After harvesting, the algal biomass can be processed for various uses as we need it for our daily life. The first step is drying. Algae can be dried by using various methods like spray drying, freeze drying, and sun drying. And then extraction. Available compounds present in the algae such as lipids, proteins, carbohydrates, and pigment are extracted for the therapeutic uses. This can be done by using various solvents as well as mechanical methods. And then fermentation. Some algal species such as spirulina and chlorella can be fermented to produce higher valuable compounds. Thank you, one and all. Hello everyone. Today we are going to